All right, good morning, everyone. So just to give you a heads up, so we have our meetings this week. I'll be meeting with the first team tomorrow and then the other team on Friday. So some of the things that we're going to discuss are the organization of ideas based on what I've seen in the annotated bibliographies, in addition to some tips on how to present the material, how to cite the references, and also about figure making and even taking figures from the literature. So we'll be talking about that in our specific meetings. On Thursday, there is an optional lecture, which I recommend those, especially those who are focusing more on the structural components of cytochrome C, to attend, because it'll be really nice to make your own figures of the protein with respect to whatever component that you're going to be focusing on in your review. So that workshop is going to be offered by Josue Benjamin. So it's basically using PyMol and taking structures from the protein database and be able to produce their own figures. All right, so that will be Thursday during the class time. It is optional, but I do highly recommend it. All right, so what we're going to do is transition to discussions that look at immunity and as it relates to metals. And then in the lecture next week, there are a few presentations that are going to look at this with a little bit more depth. All right, so I'm going to go ahead and project. All right, so what I would like to start off is giving you a little bit of the basics about immunity. And so just to give us a perspective as to how that really plays a role in terms of what impact certain metals can have in the body. All right, so a lot of this material is quite basic. It does come from, I would say, introductory biochemistry, but I wanted to give you this context so that you'll be able to see this system that we do have in place as a defense mechanism. So the human body has two lines of defense, primarily the innate immune system and the adaptive immune system. So as you can see, the name implies one, both of them serve as a way to defend us against pathogens. One is basically a more general response system to some general features present in pathogens, while the other one is more of it adapts to different components that become present from a given pathogen. So the innate system responds rapidly to features present in many pathogens, so it's a common structural components, whereas the adaptive immune system responds to specific features present only in a given pathogen. And the idea is that they both work to eliminate or neutralize those pathogens. Now, here is the major challenge in the identification of pathogens, and that is to produce a system of receptors diverse enough to recognize a wide array of potential pathogens, but also to distinguish invaders and their disease-causing products from the organism, the host's own products. And so that is a situation of being able to distinguish between the self and the non-self. So when we're talking about the innate immunity, so this is evolutionarily an ancient defense system. It's found in, at least in some form, in all multicellular plants and animals. It is the frontline defense mechanism against foreign pathogens and basically relies on the recognition of common features of invading organisms. So by common features, for instance, we're talking about well-conserved specific glycolipids or nucleic acids that, in theory, are absent in the host cell or organism. Again, the distinguishing features between the invading pathogen and the self. So as far as the components of the innate system, this includes the epithelial lining that surrounds host cells, specialized cells called phagocytes that can ingest and destroy pathogens. So one of the major proteins that really participates in this innate immunity, the toll-like receptors, or TLRs, they are among different family of receptors that are expressed on antigen-presenting sentinel cells. So examples of these type of cells are the macrophages and then the dendritic cells. And so essentially what they do is they recognize specific features present in most pathogens. These can be found on the cell membrane. Okay, so it's essentially recognizing things exterior to the cellular environment, or they can be found on the internal organelle. So imagine a pathogen that enters into the cellular environment. So these are able to then, initiate a immune response when recognizing them within the actual cell. And so the toll-like receptors have a common structure, as depicted in this image here. And one of the major features is the presence of a large extracellular domain with 18 to 27 leucine-rich repeats. So that is depicted here in these blue, I guess you can call these rectangular-like representations, although that's not actually how the structure looks. So each of these repeats consists of 20 to 30 residues, of which 6 of those residues are leucine. Then the next component is a cap, that is a cysteine-rich domain. Then you have a single transmembrane helix, represented there, and then an intracellular signaling domain that serves as the docking site for other proteins. So as you can see right here, signaling domain. All right, so let's look a little bit at the leucine-rich repeat. All right, here we have a better perspective of what that component of the toll-like receptors looks like. So here is a representation of that extracellular domain. So the leucine-rich repeat units come together to form a central parallel beta sheet that curls to form this concave structure. So you have the beta sheets. You see this kind of structure right here, which is concave. Okay, and then when we look at one specific repeat, as represented here, okay, you can see the presence of 6 leucines. So essentially what you have is within the single LRR unit, the leucine residues form a hydrophobic core, and that's actually quite important. All right, and that's important because of the following. It is the pathogen-associated molecular pattern, or the PAMP. The PAMPs are the specific molecular characteristics that each toll-like receptor targets on invading organisms. Typically, a critical component of the pathogen that cannot be readily mutated because that mutation would affect pathogen function. And so here I provide a table of certain PAMPs that are recognized by specific receptors. I'm just going to highlight a few of these. So for instance, here you get the receptor, you get the localization, the PAMP, and then what is believed to be the source of that particular PAMP. So for instance, the toll-like receptor 1, localized on the plasma membrane, has the capacity to recognize triacyllipoproteins, whereas, for instance, the toll-like receptor 3, which is found on the endolysosome, has the capacity of recognizing double-strand RNA. Right? So we see these 2 distinguishing features, and so for the source of the PAMP for the toll-like receptor 1 predominantly comes from bacteria, where the double-stranded RNA comes from viruses. So here we get a structural representation of the interaction of a toll-like receptor with one of the PAMPs. So as we can see right here, this is the recognition of the PAMP that corresponds to the toll-like receptor 3. So recall, that particular receptor has the ability to recognize double-stranded RNA. So we see the interaction of the double-stranded RNA interacting with the leucine-rich repeats. And so in this particular representation, we have this dimerization that enables that interaction between the RNA and the leucine-rich receptor. Sorry, it's leucine-rich repeat. Excuse me. All right, so that is just a generalized discussion of the innate immunity. Let's talk a bit about the adaptive immunity. And so here we're talking about 2 parallel but interrelated systems, the humoral and cellular immune response. All right, so the humoral immune response, so this consists of soluble proteins called antibodies, so essentially immunoglobulins, function as recognition elements that bind to foreign molecules and serve as markers signaling that there is a foreign invasion. And these are secreted by plasma cells, which are derived from, so plasma or blood cells, which are derived from or beta lymphocytes, or B cells. So a foreign macromolecule that binds selectively to an antibody is called an antigen. The specific affinity is not necessarily for the entire macromolecular antigen, but for a particular site on the antigen called the epitope. Each B cell produces just one type of antibody that can recognize a single epitope. And it turns out that the immune system generates more than 10 to the 8 different antibody proteins. So here I present to you an overview of the immunoglobulin classes. And so just some of the distinguished structural features and their relative abundance within serum. So these are, again, are proteins that are produced by the blood cells. So you have the immunoglobulin G, A, M, D, E, right? They are present at different concentrations. One of the overall consistent feature of these particular proteins is the presence of a common light chain. So as you can see in this figure below, that gives us sort of a visual representation of the different immunoglobulin proteins. The light chain, which is depicted in yellow, is a common feature in all of these. But then the distinguishing feature is the heavy chain that's present. And so you have just a representation of the immunoglobulin G, A, M, D, and E, and just to give us a perspective of what these proteins essentially constitute. And another important thing is, at least for the higher order structure proteins, is that their quaternary structure is stabilized by the presence of disulfide bonds, and these are actually represented by these green bonds. Okay, excuse me, I'm not sure why that keeps happening. Okay, so to talk a little bit about the cellular immune response here. So you have cytotoxic T lymphocytes. So these are also known as killer T cells that destroy cells that have been invaded by a pathogen. And they do so by recognizing markers on the cells. So the cellular immune response is mediated by specific receptors that are expressed on the surfaces of T cells. Because intracellular pathogens do not leave markings on the exteriors of infected cells, vertebrates have evolved a mechanism to mark the exterior of cells with a sample of interior contents, both self and foreign. So here we have just a representation of this recognition here. So here, some of the internal proteins are broken into peptides, which are then bound to a complex of integral membrane proteins encoded by the major histocompatibility complex, abbreviated as MHC. And so here we have a figure that gives us a perspective of this recognition. So we have the presence of an MHC to which is bound a peptide. And that peptide is essentially serving as a marker, tagging the cell. And so that marker is then recognized by a T cell. So here we have the depiction of a T cell interacting and the receptor interacting with this complex that has that peptide marker present there. And so that interaction essentially calls for the activation of the T cell. And there is as far as the signaling goes, there is a subsequent downstream phosphorylation series of events that occur. The immune system generates more than 10 to the 12 T cell receptors, and there are helper T lymphocytes. These helper T cells contribute to both the humoral and also the cellular immune responses by stimulating the differentiation and proliferation of both the B cells and also the T cells. It turns out that the T cells are subjected to what are known as positive and negative selection. So these are processes in the maturation of these particular cells that occurs in the thymus. So during the maturation process, the T cells from the thymus are first subjected to what is known as a positive selection. So in this process, those cells for which the T cell receptor can bind with reasonable affinity to either class 1 or class 2 MHC molecules survive the selection. The remainder undergo apoptosis. Then following that filtration process, then the cells undergo the negative selection. So then here, T cells that bind with high affinity to the MHC complexes bound to self-peptides expressed on the surfaces of antigen-presenting cells in the thymus undergo apoptosis. So those with moderate affinity will be able to complete maturation. So the importance of the negative selection is to be able to minimize recognition of components of the self, and that is to avoid a process that is known as an autoimmunity. All right, so what exactly is an autoimmune disease? So that occurs from the generation of an immune response to self-antigens. So this occurs even despite the thymic selection process, both the positive selection followed by the negative selection. Well, failures do occur. And so you do have these autoimmunity diseases that can develop. One of the examples of an autoimmune disease we have mentioned previously, for instance, the insulin-dependent diabetes mellitus. There's also multiple sclerosis and rheumatoid arthritis. So these type of diseases result in damage to selective tissue that express the antigens that are recognized by the immune system. In many cases, the causes are unclear. So, for instance, there are examples where infectious organisms such as bacteria and viruses may play a role in the onset of these type of autoimmune diseases. So, as an example, an infection may lead to the generation of antibodies and T cells that react with different epitopes from the infectious organism. Now imagine that one of those antigens closely resembles a self-antigen. So because of that close proximity to the structural features of that self-antigen, then an autoimmune response can result. So what I would like to discuss is, and this is presented in this article here, is those autoimmunities that can actually be stimulated by the presence of heavy metals that can enter into the body. So as we know, metals can play a very beneficial role and also detrimental role with respect to the immune system. And so when we're looking at metals as potential invaders, so we know that metals, obviously, they're not necessarily pathogens in the what we would consider in the traditional sense, but their toxicity related to certain metals can be in the form of it can lead to a form of autoimmunity. So several transition metals, such as mercury, silver, and gold, have the capacity to induce changes in cells of the immune system, of which mercury is the metal that is most known to trigger an autoimmune effect. Now, talking a bit about the mercury, it is a ubiquitous metal. So ubiquitous meaning that it's highly prevalent in our world, in the environment. It is a major pollutant, but it's also used in biomedical applications. So it's found in dental amalgams, cosmetics, vaccine preparations. At least to a large extent, it was used heavily in biomedical applications, much less now. Can be found in preservatives and fumigants. And so indicators of high exposure to mercury in the body are the following. Examples of elevated T cells, elevated immunoglobulin levels, such as the IgE. Some people produce anti-nucleolar antibodies. So the nucleolus is the largest structure in the nucleus of eukaryotic cells, which is important for ribosome biogenesis. These antibodies form due to recognition of a marked nucleolus as foreign. All right, so I want to ask you, what do you think then is actually happening? Why would a particular component of the body, or in this case of the cell, be marked as foreign in the context of a metal? What do you guys think? how can the presence of a metal make something that is innate to us appear as foreign? Anyone? You can put your thoughts in the chat room, or you can get on the microphone. Right. So perhaps some sort of metallation or extensive metallation, I would say, is resulting in a structural change such that, yes, many of these features no longer have their native form, and so they would be no longer recognized as part of the body, or potentially would be just seen as something that's foreign. Yes, I believe that would be the correct thinking here. All right, so it turns out that there are certain rodent models that can help to give some insight into this metal-induced autoimmunity. So some, but not all rodents exhibit a metal-induced autoimmunity. Studying genetically susceptible rodent models allows insight into how metals can induce autoimmunity, important considering how many metallic materials are actually introduced into a human body. So as I already mentioned, there are certain dental applications that do use metals. In addition, as I've mentioned previously, there are also metals that are introduced in the form of implants, prosthetics. So a type of rodent that is susceptible is a brown Norway rat. Exhibits autoimmunity when dosed with subtoxic amounts of mercury chloride. So this is an amount of 1 milligram of the metal per kilogram of body weight of the animal. So what has been observed is the following. Similar results, such as production of antibodies. So the antibodies are in the form of double-stranded DNA, anti-phospholipids, anti-glomerular basement membrane of the kidney. You also see the polyclonal activation of B and T cells. So a single antigen is recognized and attacked through its epitopes by multiple cells. You also have renal immune complex deposition, resulting in this condition, which is an inflammation of the kidney. Right? So you see many of the same types of features that appear in humans. But another really interesting thing is that over time, these particular genetically susceptible rats can become resistant to the effect of the mercury. There are also certain mice that are genetically susceptible. For instance, lupus-prone mice. So I'm not giving you a specific type of mice here, or mice model. The mice models exhibit similar consequences as rats. For instance, you have the autoantibodies that form, although in this context seem to be restricted to the nucleolus. The main target of the autoantibodies, this fibrillarin, a 34 kilodalton ribonucleoprotein that is associated with several small nucleolar RNAs. All right, so mechanisms of metal-induced autoimmunity. So the autoimmunity response is part of the adaptive immune system. Mercury can affect the expression of various genes in immune cells through subtoxic dosages. Those subtoxic dosages are administered. There may be localized toxicity. So for instance, mercury-induced cell death can result in a 19 kilodalton cleavage fragment of that protein, the fibrillarin. All right, so these are additional things that the mercury is able to induce, in addition to the things that you guys have proposed, that there is metallation, but also at the level of affecting expression of genes, even causing cell death. Other proposed mechanisms, for instance, mercury 1 and 2, and even gold 1 and silver 1, have a very strong affinity for thiol groups. Right? So we know the properties of these metals as being soft, and so they have this capacity of being able to bind to soft base groups that are present in biomolecules. Mercury 1 and 2 bind to glutathione, which is, as you know, the most abundant source of intracellular thiol. So as a consequence of that interaction, it can cause a redox imbalance. It can also inhibit glutathione regulation of this cannabidiol A mediated induction of the cytochrome interferon gamma. So the cytochromes are a family of proteins that bind to specific receptors to stimulate B and T cell growth and differentiation. Can cause a shift in cytochrome expression profiles. And mercury exposure can dramatically worsen autoimmune manifestations. All right, so here we're going to look at the perspective of metals in the treatment of autoimmune disease. And so there's actually a little bit of a complexity here, and I'll emphasize that at the end of this discussion. So here we're going to look at the application in rheumatoid arthritis. This is a systemic autoimmune disease in which the body attacks the lining in the joints and the fluid in between. It results in the erosion of opposing bones and leads to severe impairment of movement. And so traditionally, I would say even today, treatment is with pain-killing drugs such as aspirin. Also, disease-modifying arthritis rheumatoid drugs, abbreviated as DMARDs. These are immunosuppressive drugs. One of the really important ones is Humira, that is used commonly today. And so for the longest time, cryotherapy was in development and was applied for the treatment of rheumatoid arthritis. So this is the use of gold compounds, and I've introduced this previously in the context of gold applications for anti-cancer therapy. So this cryotherapy was a phrase that was coined by the French physician Jacques Forestier. So here, gold thiolate compounds were used for this application, as they were quite effective. So here we have the structural representations of these particular gold thiol compounds, which you actually have seen previously when I introduced gold compounds. So these three, sogenol, myocrystin, sanocrystin. Again, these are the gold thiolate compounds. One of the lead compounds was aranafin. So it was developed as an orally deliverable drug. And essentially, it reduced the nephrotoxicity, so this kidney-related toxicity, relative to other gold thiolate compounds. And that is because of lesser excretion via kidneys, also reduced gold retention. Remember, I briefly discussed about that issue of the toxicity due to gold being retained within the kidneys. So this was a drug that was released in 1985, although it was actually not particularly used for the application in the treatment of rheumatoid arthritis. And so here are just reminders about the structure of this compound. So we have the thiolate component that comes from this thiolated acetylated glucose, and then you have the phosphine group, this triethylphosphine. All right, so I want to talk a bit about the biodistribution of aranafin. And there's also, as we will see, it's not entirely clear how the aranafin manifests its effect in a way that can serve as a sort of immunosuppressant drug. But some of the things to consider when understanding what effect this compound actually has. So it is actually not very water soluble. So to be able to solubilize it, to an extent, you have to possibly use a co-solvent to be able to do so. And another really interesting thing is that the effects of this compound are slow to become apparent. In some cases, at least 1 year was required of usage of this compound in order to see an effect. And that is because of a slow accumulation of potentially the metal throughout the body. So to get a little bit of perspective about this, we have to understand the sort of biological fate of the metal, of the compound. And so we can look at this from the perspective of clearance from the body. All right, so that's one way of gauging sort of the pharmacokinetics, the pharmacodynamics of a particular drug. So here we're looking at a study that was done in which isotopically labeled forms of the compound was prepared. So we have sulfur 35, gold 195, and the phosphorus 32. And so as it turns out, when you look at it from the perspective of these components, the phosphorus can be cleared from the body within 8 hours. And so we see this biotransformed form of the phosphorus. The sulfur component of it has a half-life of about 16 hours. Okay, so we're looking at this in terms of half-lives. And then the gold component has a half-life of about 20 days. So very clearly, we see a different profile for all three, the metal and the two ligands. And so what this suggests is, to an extent, that that perhaps the retaining the ligation is perhaps not necessarily the important feature of this particular drug. Now, that is to an extent in contrast to what we discussed about the application of Aranafin for an anti-cancer application, where we focus on at least a certain feature of the compound being important for the delivery of the gold into the cellular environment and for it to have an effect. All right, so let's talk a bit about this biological fate, but from the perspective of a sort of in vivo stomach speciation. So to mimic the pH of the stomach, the degradation of the compound was examined at a pH of 1.5 in the presence of a high concentration of salt. So typically salts are included to give an ionic strength that's fairly high. That's to mimic the ionic strength of the body, of physiological fluid. So this type of study was done using stop flow kinetics and UV-vis detection. All right, so essentially, to mimic this sort of degradation, we can look at it from the perspective of the compound in the presence of acidity as supplied by HCl. And so what we see in this very first step, this very first fast step, is an association of a chloro group onto the metal center. And that process of association leads to then dissociation of the thioglucose. And so now you have a rearrangement of the compound such that you no longer have the thioglucose component, you only retain the gold phosphine component of that compound. And so this is observed at this acidic pH. So this is a rapid degradation of the complex, only retaining the phosphine component. So about 25% of the oral dose is absorbed from the gastrointestinal tract for distribution throughout the body. And so part of what's contributing to that 25% is this degradation of the compound under acidic conditions. All right, so then let's consider the compound if it were to be subjected to a pH environment of the blood, and so a pH of 7.4. And if we look at it from this context, but also considering human serum albumin and what role it can play in terms of the speciation of this compound. So it turns out that human serum albumin has the capacity to react very strongly with Aranafin. And so here are just some of the features of this protein. As I mentioned previously, it is the most abundant blood protein. It is about 66 kilodalton. It consists of 17 disulfide bridges. It has one free cysteine. And it turns out that free cysteine actually serves as a very effective gold binding site within the protein. So when that complex interacts with human serum albumin, there is a release of the thioglucose component of that compound. And so essentially, you have the loss of the sugar, so minus the sugar, and then you get the formation of a compound where the gold phosphine is coordinated to the cysteine residue from the protein. All right, so this is something that has been studied at a pH that mimics the pH of the blood. And so this was done under pseudo first order conditions. So essentially, we're looking at a reaction between the compound and a high amount of the protein, leading to the observed rate constant of 0.48 per second. Or in other words, looking at this complexation that occurs with a half-life of about 16 hours. Another really important contribution or potential contribution to the speciation is the presence of a cyanate in the blood. So there's actually a a small amount that can affect the composition of aranophen. So considering the uh, the binding constant or the formation constant of gold with this cyanoligand, we have a log beta of 36.6. So that affinity is sufficiently high enough that the small amount that is present, let's say in the environment of blood, can potentially result in complete dissociation of the thiol sugar and also the phosphine group, such that then it can fully bind to the gold. And so then that complex, that gold cyano complex, can then go on to react with human serum albumin. So there is it still retains that interaction. And so there, that complex between the protein and the gold cyano complex, well, that can lead to release of the ligand. And here, a very interesting thing can occur where the metal can become oxidized, and so it can become oxidized from gold 1 to gold 3, which, if delivered into the cellular environment, that can then lead to interactions with glutathione, leading to the reformation of a gold 1 species. All right, so from either context, you do have the capacity of the gold as gold 1 being able to enter into the cellular environment. All right, so the reason I'm presenting this and why I'm placing an emphasis here is because I want to make a distinction between this biological fate versus what we talked about when we were talking about the effect of varanophen as far as anti-cancer activity. So it's believed that for the effect that the gold can have in terms of a rheumatoid arthritis effect, well, it appears that it's very important for the gold to be able to biodistribute to the body. And to facilitate this, it is preferred that the gold does undergo this dissociation of its ligands and it affiliates with human serum albumin, which can potentially prolong its existence within the blood and potentially enable its biodistribution within the body. So, as far as from the perspective of the importance of this compound, it appears that really it's the gold component that is the most important, and the prevalence of it in the body and its affiliation with the human serum albumin is key to enable that biodistribution. That is in contrast to what we talked about as far as the application of varanophen for an anti-cancer application, where you do want to minimize how much, I guess you would say, dissociation of this complex occurred, because you want to retain a certain component of it so that it can actually have a relatively fast uptake into cancer cells so that the gold is able to reach its targets. All right, so that's two different, I would say, two different biological fates. One enabling one type of effect, whereas the other enabling another type of effect. So one is this sort of slower release of the gold and a longer retention to have this rheumatoid arthritis effect, and one where you want this rapid delivery of the gold to a cancer cell to enable that effect.
Professor, I have a question.
Yeah, go ahead.
On slide 29, like 28 and 29, when
Yeah.
you say that 25% of the oral dose is absorbed, that's absorbed before reaching the gut, the stomach.
So that's absorbed into the gastrointestinal tract. Okay, so that's going to be there's going to be what goes into the acidic fluid, and then whatever amount can actually be absorbed into those cells, and then delivered into the bloodstream. Only 25% of that actually gets absorbed. What that composition exactly is, so I would say it's mostly the 25% in terms of perspective of gold, because we don't actually fully know. It would be hard to predict exactly, if that's 25% of the whole compound, but really 25% of gold species that's able to be absorbed into the gastrointestinal tract.
So, but the two products that like are produced, the thiol sugar and the complex with the phosphorus, I don't understand how, if like you have the gold and the phosphorus complex and the half-life of the gold is 20 days and the half-life of the phosphorus is 8
Mhm.
hours. So at what point do they actually dissociate so that you can excrete one before the other?
Right. So you have a significant amount of dissociation within just the environment of the stomach. And then within the bloodstream, it appears that that actually can go even faster if you consider, we're only just talking about two major components here. We're not even considering all the constituents of blood. But if you factor in human serum albumin, and you also factor in this small anion, this cyanide, right? Those are sufficient to result in loss of both of the ligands and also sufficient to enable the gold to be bound by the albumin. So either pathway can result in further release of ligand. The one to the right can result in complete release of the ligand. So if we look back, let's see, just ignore my notes here. If we look back to here, right, the the goat is the one that has the longest retention in the body. These other two, it's a little bit hard to understand with why one is necessarily longer lived than the other. There could be a number of factors in terms of filtration of the of the thiol sugar and the filtration of the phosphine group. But really, it is the gold that has the longest retention. And for that to be possible, there has to be some sort of biomolecular interaction with that metal to enable for it to be able to stay in the body. And so what's believed to be the case is that the interaction with albumin allows for that prolonged existence, in addition to just it being distributed throughout the body, potentially through the use of albumin serving as that vehicle for that gold. But yeah, so the main thing is to consider, what are those factors that are contributing to dissociation of ligands? So clearly, the acidity in the stomach is enough to contribute to a great dissociation of the thiol group. But then also within the environment of the blood, the presence of other gold buying groups are sufficient to be able to get rid of both the phosphine and also the thiol sugar, even to a greater extent. Because that's the way we're supposed to consider it, is those factors, whether it's a small molecule or whether it's these big proteins that have the capacity to affect the speciation of that gold, and what's going to lead to its I would say its greater lifetime within the body. Does that answer your question?
Yes, that I got a little lost, but you explained it. That's why I asked. But yeah, thank you.
You're welcome. Okay, so excuse me. All right, so due to the slow nature of the effect of auranopin and its accumulation in the body, elucidating the mechanism of action of gold anti-arthritic drugs is difficult. And so I will say that what I'm mostly going to show you in the remaining slides, and they're really not that many, are what to an extent we already know some of the where some of that metal can localize, but it's not entirely clear for some of these interactions how that can possibly be related to this anti-rheumatoid arthritis effect. So in these type of studies that have led to this sort of identification of gold metallation of certain biomolecules have been done by targeted approaches. So as I mentioned previously, targeted approaches typically are that you take a molecule and you can see that if it has any binding interaction with a certain metal. All right, so again, those type of studies have been done to determine which biomolecules may interact with the drug or its proposed biotransformation products, right? Because as you can see, that compound does not remain quite stable, right? So really, it's possibly some of these biotransformed products that are probably a more important model of how that metal may actually localize. And so how this interaction may alter the activity of the biomolecule. Most likely targets of the gold are proteins and thiol or thiol ether containing small molecules. So one example of a target is the cathepsins. These are a group of cysteine proteases that hydrolyze elastin in cartilage and collagen in bone, believed to play an important role in the resorption, degrading of bone. So these enzymes are found inside lysosomes in osteoclast cells where gold accumulates. So it's found that the gold binds to cysteine 25 residue of cathepsin K, and a residue important for enzyme catalysis. So at least this interaction, to an extent, can lead towards an understanding of how gold might serve as a therapeutic role for this particular application, because by inhibiting these type of enzymes that can result in degradation of bone, that can serve as a form of treatment. What's not so clear are some of the other targets. So as we mentioned previously, the thyroreducin reductase system is a very important one, as we've seen for the perspective of an anti-cancer application, but it's also believed to be correlated to an extent with the effect that can be induced by the gold. And so here, as we know, the gold can bind to the active site, the selenium, cysteine, or cysteine residues of thyroreducin reductase and the thyroreducin proteins. We know that these proteins are responsible for redox balance. They play an antioxidant defense role. They also play a role in terms of nucleotide metabolism. The gold can also bind to a related protein, the human glutathione reductase. It can actually bind to the cysteine 58 and cysteine 63 residues of this protein, although the correlation of its in vivo relevance is not so clear. However, this interaction with the thyroreducin reductase system is not entirely clear how it necessarily plays a role here. Perhaps it can trigger an immune response that enables sort of a therapeutic role here, but that's not entirely clear how that really can contribute to, something that is already an autoimmune disease. That's not a clear finding. You also have the finding of gold interacting with the protein tyrosine phosphatases. Okay, so we've talked about these type of enzymes, at least in the context of vanadium playing a role with inhibiting a specific phosphatase. Well, these are cell signaling proteins, and gold has a capacity of binding to the active site cysteine 227 of the lymphoid-specific tyrosine phosphatase, and also has a capacity of binding to nearby cysteine residues, for instance, cysteine 129, cysteine 231. But this interaction is not entirely clear what effect that it has, and as I was saying in a moment, maybe it's actually not necessarily an interaction that is actually favorable. Gold also has a capacity of interacting with the zinc finger proteins, and we actually have seen this type of effect when we talked about platinum-2. So in this context, zinc finger proteins, as I previously mentioned, they are very important proteins participating in a number of cellular processes. So in this context, the gold can displace the zinc from these proteins. So this is a form of transmetallation. And so what you can essentially form are gold fingers. So basically the gold substituted form of the zinc fingers. So they can bind to the different zinc finger peptide motifs. So these are the typical motifs that you find that can coordinate to a zinc, and obviously the gold one can substitute for the zinc in this context. And so a really pronounced feature of gold interacting with cysteine is the formation of this strong ligand metal charge transfer band that appears at 310 nanometers. Again, this substitution is not clear whether this is a favorable thing. It most likely is not a favorable thing, given the importance of these zinc finger proteins, and the gold substituting for them may not actually enable some of the properties that the zinc version can fulfill. And so this is where I was mentioning to you why it's so complex to understand what the gold is actually doing, because the gold itself, and as I already mentioned earlier, the gold itself can actually complicate the symptoms of rheumatoid arthritis, given that gold has its own capacity of triggering an autoimmune condition. And so it's very likely that some of this, I would say, off-target effect of metallating other proteins can, in the long run, lead to the formation of an autoimmune disease, making the application of gold not necessarily a favorable one, at least not in the context of the use of this particular type of drug. So as I mentioned previously, Orangutan is something that's no longer applied for this application, perhaps because the compound is not sufficiently stable enough and is relying on release of the gold and distribution via albumin, and that can lead to simply a lot of off-target binding that ultimately can result in autoimmune disease or worsening the symptoms of the rheumatoid arthritis. So it's interesting that it takes so long to have an effect, and who knows how effective it was even when it reached that point. So that just gives us some of the complexities of this metal, which does have, to an extent, an immunosuppressive role, but does have the capacity to then trigger its own autoimmunity. All right, so I'm glad Adriana brought up that question, because it is a little bit confusing, if to think about how this drug really has an effect of this nature, at least from the perspective of the anti-cancer application. There we could see where the targeting of this metal to a particular redox system can induce a response that leads to cell death. But in this context, it's not so clear how the gold, when it reaches certain cellular targets, really can enable its effect. All right. So I will leave you with that. Okay. If you have any additional questions, please let me know. So again, I will be meeting with both teams, one on Wednesday, one on Friday, and then we have our optional lecture on Thursday to give you some insight on drawing structures, protein structures, using the program PyMol. All right, everyone, have a good day, and I'll see you later this week.